Here we have the quick guide to Laboratory Quiz 1. I think that making a short video on some of the important areas might help you concentrate your studying. As usual, anything in Fox or the supplement, or for that matter, the videos or the slides is fair game, but this guide has been established to help you concentrate the areas where you think you can best spend your time to get the most, bang for your buck in terms of studying. I will pull at least one question from the back of Fox or the supplement for the quiz, literally word for word. And the information for the quiz will be posted as an announcement and also on Canvas in the quiz module. So look for it there. So let's go through the quiz. Laboratory Quiz 1 is on labs 1, 2, 3, 4, and 6. Remember, there is no laboratory 5, so it's on the first 5 laboratories that we do. So let's kind of look at laboratory 1 first. The most important thing from this lab are the calculations in the supplement. The guide says students must use their own calculators for this purpose. However, since we're not in class, obviously, hopefully you have an access to a calculator that you can use. You did the calculator portion of the quiz, so that should provide you with enough information to be able to do any of the calculations in class. It's just a series of those calculator operations, and you just have to make sure you put them together in the right order. All right, so some of the more important aspects: you need to be able to convert within the metric system for length, weight, and volume. All right, so temperature is excluded. Don't worry about temperature. And I just want you to be able to do it for the common metric units that we use in laboratory. Things that would appear in laboratory would be things like kilo, centi, milli, and micro. While some medical tests might measure things in pico or nanograms, but for the most part, those are the big 4. So you need to be able to convert those fairly readily. So if I said, Joe is 32 centimeters tall, how many millimeters is Joe, or something like that. If Joe's only 32 centimeters tall, Joe's probably very young. But being able to convert within the metric system is very important from that. I also expect you to be able to go from the metric system to our system and back for height and weight. So a good exercise is to convert your height and weight to the metric system. And if you're familiar with the metric system for your height and weight, then go the other way and convert it to inches and pounds. If you're asked to do this on the exam, you will get a sheet that will have the conversion factors on it for the information between the metric system. You will not get a sheet with the difference between kilo, centi, milli, and micro. I expect you to know that. You'll also have to be able to calculate the mean and standard deviation. I can tell you for sure there will be a question on mean and standard deviation. You'll get a number of results from some sort of experiment or something, and from those you'll have to be asked by hand to be able to calculate the mean or standard deviation. You need to be able to show me step by step how you did it. You need to be able to show me what the correct answer is as well. There are 2 formulas you can use. They're both published in the lab supplement. You can choose either one. Take the one you're most comfortable with and practice that one. There is a practice set of problems for lab 1. If you go to lab 1 page in the module, and you can download that if you haven't already done so. And one of those problems is a standard deviation and mean problem. It'll be worth 2 points for each. So the question itself is worth 4 points, with 2 points for mean and 2 points for standard deviation. So if you stop and think about it, right, your total exam is 25 points, and you already know what 4 points of it are. So you have almost 20% of your exam. You already know what the question will be for that. For minor importance, you should be able to understand ratios, dimensional analysis, how to figure out unknown values and things like that. The 2 problems in the supplement, help with that, but I think that should be sufficient knowledge for the exam, and even then, it'd probably be a little bit of overkill, to be honest. Of minor importance, the percentages for the white blood cell and frequency distribution. I'm not going to worry about that right now, because later on this semester in lab, we're going to do a differential white blood cell count, actually. So why would I test you on it now if we're actually going to do it and incorporate it into some lab? Of minor importance is how to effectively prepare solutions in a practical sense. So the molarity and things like that, we're not going to worry about yet. Again, in a future lab, we're going to worry about calculating molarity from concentrations and then using that to calculate osmolarity. So for those, we're not going to worry about those either, because again, we're going to do it later and you'll get better at it. And if we don't do it later, it's probably not that important to worry about now. So don't worry about those things. As I've pointed out in an announcement and on those online sessions, you are not responsible for FOX 1.1 and 1.2 on laboratory exam 1. If you carefully read the supplement, it says review FOX 1.1, 1.2. When it says review in the supplement, it means you're not responsible for it on a laboratory exam. Okay. However, the dimensional analysis may be useful for you if you're dealing with especially metric conversions. But if you know how to do conversions already, then you don't need to go look at that. You will not be asked to make a percent solution on an exam, and I mentioned this in one of the videos. Percent solutions are really easy to make, but their functional properties are very difficult to understand, and they're not very useful. So making a 10% solution of sodium chloride or glucose really isn't useful functionally, so we don't worry about it. And again, I mentioned this, why it's good to know in practice at this time, you're not responsible for converting solutions of glucose and sodium chloride to milligrams per deciliter, then millimoles, and then milliequivalents, and then milliosmoles for this exam. Again, in a future laboratory, we're going to do that, so I'm going to test you on those things for laboratory exam 2. So don't worry about them here. So really, when it boils down to what do you need to know and study for the first laboratory, it's conversions within the metric system just for kilo, centi, milli, micro. Conversions between the metric system and our system just for height and weight, and the mean and standard deviation. I don't know exactly how many points are on your lab quiz that's represented by lab 1, but I'll tell you this, I know it's more than 4, and I already know there's 4 points for mean and standard deviation, so it will help to learn those things and study it. All right, for laboratory 2 had 2 sections of FOX that you're responsible for, 1.3 and 8.4. When we have sections of FOX that we're responsible for, and this is true for the whole semester, you are responsible for the background information as it relates to the lab that we did, the information in FOX, the little clinical boxes. Okay, so when it talks about, diseases or applications, you should read those. I do, to be honest, so I have no problem making that. When FOX gives ranges for different things, you should know those as an example. And then the questions at the back, except for the clinical investigation questions, those are pretty difficult, and I wouldn't ask one of those on an exam. All of those answers are posted So your job is to look at the posted answers once you've looked at them, and if you have a question about why an answer is so, then ask your lab partner. We have the tutoring schedule that's up, right? So that's posted in the announcements on Canvas, so you have access to the tutors, or you can ask me. So you have a lot of resources there if you don't understand something, but helping yourself do that, right, is a good thing. So, look at those and get those done. So specifically for FOX, we did 1.3 in Lab 2 and 8.4. So 1.3 had two parts. The first part was the water bath and how the water bath works. And so the information about the water bath, where it talks about the sensor and the integrator and things like that, and how it works through negative feedback, right? So you have it set at a certain temperature where when it reaches that temperature, the heater goes off and we measure the temperature, and then we take the average and say, okay, that's the set point, right? Once we have that set point established, we dump ice water, some sort of cold water, to drop the temperature, and that kicks the heater back on and heats the water back up to the temperature again. And that process is a great example of negative feedback. And so doing that would show negative feedback. We had a set point, we went away from the set point, and we brought it back to the set point. However, it's not a good example as an example of homeostasis. To have homeostasis, you need antagonistic control. So our water bath, if we dumped boiling water into it and made it hotter, we don't have the ability to cool it off. And to make it a better example of homeostasis, it still wouldn't quite be there, but it'd be better if it had antagonistic control. So if we had a heater and some sort of cooling mechanism, that would make it a better example, because then we'd have antagonistic control. In 1.3 of FOX, we looked at heart rates. And one of the things, despite what the title says, is heart rates are not homeostatically regulated. Okay? We actually use heart rate to control blood pressure. It's called mean arterial pressure, the average blood pressure over time. There are many variables that go into the control of heart rate, and we'll mention a few for resting heart rate in a minute. So one of the points of the lab was to demonstrate that people who regularly exercise, on average, generally have a lower resting heart rate than people who don't. And that's because their heart is bigger and stronger, and basically they can pump more blood each beat. And scientifically, the important concept is they have what's called a greater stroke volume. The need for oxygen for someone who's physically fit isn't any different at rest for somebody of similar size who isn't as physically fit. So if you have a stronger heart where you pump more blood, then you need less beats to pump that blood through the body. Exercise capacity would be different, but the resting pulse for those people, because people that regularly exercisers in general have a bigger, stronger heart because they can have a greater stroke volume, they need less beats to accomplish that. But heart rates are extremely individual. They're determined by other factors besides fitness: genetics, body size, gender, and a number of other issues play a significant role in determining that. And so, you could have someone who was very fit and have a heart rate of 86 at rest, while someone who's very unfit and have a heart rate of 56 at rest. So you just can't use heart rate as like an end-all be-all to measure fitness, because there's a lot of variables that go into it. But on average, especially if you take a large group, the larger group with the large group that exercises, right, would have a lower average heart rate. And in fact, I would be surprised if any of you decided, okay, I'm going to start exercising, and we measured your heart rate a year from now and it hadn't gone down significantly if you exercise more than you do now. Even if you're exercising now, if you exercised harder, longer, more often, then your heart rate would likely go down at rest, because again, you're making your heart a better pump and you'll have a greater stroke volume. Body size matters within species. Smaller people tend to have faster heart rates, just like smaller animals in general tend to have faster heart rates. Women tend to have a faster heart rate than men, but we're not really exactly sure why. So if you factor out all the other things, women still tend to have a little faster heart rate than men. Often cited as hormonal differences, but I'm not sure how well that's been studied versus just assuming, since men and women have different hormones, men and women have different concentrations of hormones, we actually have the same hormones, then that obviously could play a role. The other part of FOX for number 2 is 8.4. So 8.4 had 3 parts. The first part was about buffers and how they resist changes in pH. And they neutralize the solution by complexing the free H plus or OH minus, so that doesn't affect the pH of the solution. So understand how buffers work, understand how carbon dioxide acts as an acid in the blood. That's this equation here. When you have CO2, right, that forms with water to form carbonic acid, H2CO3, and that dissociates into H plus. Remember, all acids, bases, salts tend to dissociate in water. H plus plus HCO3 minus. So we get this H plus, and that's why the pH drops. And that's an important equation actually for all of these portions of the lab, but it's important for that. In B, we looked at the change in metabolism and how that affects CO2. And the way we did that is we had people make a basic solution with an indicator called phenolphthalein. And phenolphthalein is a pink indicator in a basic solution. And what we had the person do is we had the person basically titrate acid into the solution, drop the pH, and when the pH became neutral, so it went from a base, right, so a basic pH to a neutral pH, the indicator turned from pink to clear. So what happens during that experiment actually is the way we titrate in acid is we bubble carbon dioxide in through a straw. And carbon dioxide, as we saw this equation up here, right, CO2 forms H plus and HCO3 minus, right? So the more CO2 you have, the more H plus you have, and the faster you're going to change pH. So the time to color change before and after exercise is dependent upon the amount of acid you have. So if you exercise harder, you're going to increase your metabolism. You're going to increase the amount of CO2 that you blow off. And so after exercise, we expect that time to color change to be about 50 or up to 90% of what it was originally. It depends on a lot of different factors, but it almost always works. It works pretty well. Part C is kind of looking at the same idea in some ways. And what we're looking at there is we're measuring the breathing rate. We're going to measure the breathing rate before and after hyperventilation. So the average breathing rate in humans is about 12 to 20 breaths per minute. And so the idea was, again, measure a baseline breathing rate, just like we measured resting pulse, we'll measure resting breathing. And then have the person hyperventilate. And the idea is theoretically, when you hyperventilate, you get rid of carbon dioxide. And if you get rid of carbon dioxide, it means you have less carbon dioxide and less acid in your blood. And if you have less acid and carbon dioxide in your blood, right, then you're going to have less drive to respiration. And theoretically, we expect your breathing rate to go down. All right. And so one of the things that I think you need to understand, and it's less confusing, to be honest, because we're not actually, looking at those experiments in lab this semester for the most part. When we have exam questions. The best way to answer them is what would happen theoretically unless you're told otherwise. So if I said, oh, what do you expect to happen to time to color change when you blow through a straw after exercise? And you'd say, oh, it should be less, right? So we expect that theoretically. And the same thing here. If I said, what should happen to breathing rate after hyperventilating? You should say, oh, I expect it to go down because there'll be less CO2 in the body. There'll be less acid, so there'll be less drive to ventilation. So being able to tell me that, how that works is important. But also, this one's complicated because what actually happens in most groups is people go up in their breathing rate. And the reason why is the way Fox has you hyperventilate, it's so vigorous that it actually is exercise. Moving your lungs, right, and, to inhale and exhale requires a tremendous amount of muscle movement. Your diaphragm, your external intercostals, your rectus abdominis, your external obliques, all those, your internal obliques, all those respiratory muscles, when you're breathing deeply and quickly like Fox says, it increases your metabolism. We saw in the previous experiment that if you increase your metabolism, you increase your expiration of CO2. So it's sort of like you have to balance the amount of CO2 you got rid of versus the amount of CO2 you made. And if there's a net increase in CO2, respiration will go up. If there's a net decrease in CO2, respiration will go down. And if there's no change in CO2, respiration rate will stay the same. So that's 8.4c. For lab 3 was Fox 2.1. That was looking at the plasma concentrations. And a couple notes about it. We basically understand the basic experiment, right? We took a reagent, and that reagent causes a reaction. That's what a definition of a reagent is. And so we have a reagent for glucose, we have a different reagent for cholesterol, and we have a different reagent for protein. And so if you took glucose and put it into the protein reagent, nothing will happen because it's not the right substrate. So glucose reagent will only act with glucose. And those reagents are colored. And because they're colored, they have a starting color. And again, I liken it to, because you've all done this in lab, I'm sure, at some point in time, where you had a scale in lab and you had a weighing plate, and then you push the tare button, right? That zeroes the scale, and that takes into account the actual weight of the plate itself. Okay, so you have this basic color that you need to start out with, and that's called the blank because it has the reagent and just water. There's no test substance in it. There's no glucose, there's no protein, there's no cholesterol. And so you're starting out with this base color, and you're telling the spectrophotometer, or sometimes known as a colorimeter, that this is the starting color, and anything darker than this represents absorbance. Okay, so that sets the base. Because it's got nothing in it, you call it the blank, right? That makes sense. And then we also have a tube that has the standard in it. The standard is a known concentration of a substance. We actually buy that from a biological supply company, a chemical supply company, that says we guarantee this concentration within, so many tiny percentage points. So if we know what that concentration of, let's say, glucose is, then it's called a standard because it has a standard amount of glucose. And when we take that standard amount of glucose and we measure its absorbance, and we know the absorbance of an unknown, the unknown would be the blood sample from the student in class, right? We don't know what that is. And if we have those three measurements, we could use Beer's law in order to calculate the concentration of the glucose in the student's blood, all right? So know how to do Beer's law. The general scheme of things for laboratory is you get the calculations in lab. I will give you a calculation sheet in lab, or I'll have the calculation itself on the question. And so in lab, you get them. In lecture, you won't. So I would expect you to be able to know the equations in lecture. So as an example, one of the equations that you likely think you might be responsible for in lecture would be Fick's law of diffusion. And so I would expect you to be able to know that. But for this, in lab, I will always give you the equation some way, somehow. So either an equation sheet or equation in each question. There's a couple different ways to do it. And it really depends on how the exam is being administered. So you should be able to calculate Beer's law, and then when you get the answer, you should be able to interpret that. So you should know the normal concentrations of various substances in the blood. And how do you know that? Well, there's a little box in Fox that basically shows you what they are after each little set. So in 2.1a, b, and c, right before the next section starts, there's a little box that says normal glucose is 70 to 100 milligrams per deciliter, as an example. So make sure you can determine whether it's normal or not. And I'm not going to make you responsible for the standard curve, so don't worry about it. I didn't ask anything about it. You don't have to worry about that. Lab 4 for Fox was the TLC, or what's called thin layer chromatography. Like many of the experiments we do in lab, these are more classical type experiments, or experiments that we don't do anymore. A couple of people have asked me, what's the clinical significance or the practical significance of some of these things? And the answer is, in some ways, very little, if not any. As an example, thin layer chromatography is no longer used except as a experimental device to demonstrate things in lab like we've done. But you're not going to see it in a hospital. You're not going to see it in a research lab. They have other, faster, more accurate, more sophisticated, less expensive ways to do what thin layer chromatography did in the past. However, 100-something years ago, it was useful. As an example, Banting won the Nobel Prize for his ability to sequence insulin. Insulin is a protein hormone that is made up of a specific sequence of amino acids. He used thin layer chromatography to figure out the sequence of the amino acids in insulin. So, it has some historical significance, but practically we don't use it anymore. So the amino acids are separated in a solvent, okay? And the speed at which an amino acid moves in the solvent is based on a number of different things, okay? Its polarity, its charge, its solubility, something called the hydrophobicity, and its size. In this class, we're not going to worry about all those. We're going to worry about two things. We're going to worry about solubility, and we're going to worry about how that's related to the polarity, and we're going to worry about the size of the molecule, right? So those are the only two things we have to worry about. So if two amino acids move differentially, it means at least one of those two things are going to be different, all right? Otherwise, if they weren't different, they would move the same. So for solubility, we're going to assume, which is true, that the more soluble an amino acid is in a solvent, the farther it's going to move. And what really determines solubility is the polarity. Polar molecules tend to be really soluble in polar solvents. Non-polar molecules tend to be really soluble in non-polar solvents. But generally speaking, there are some exceptions, and there's certainly a degree of each terms of that. But a non-polar amino acid would not dissolve very well in a polar solvent, and a polar amino acid would not dissolve very well in a non-polar solvent. And so those would tend to move not as far. So if I had a polar solvent, so if I had a polar solution, right, where I dissolved 2 amino acids in and they were the exact same size, but one moved much further than the other, I would say, okay, I have a polar solution, and let's say amino acid A moved a lot further than amino acid B. It would tell me that it was more soluble, right? We said they're the same size, so size isn't a factor. We've by our experimental setup, we've gotten rid of that as a possible factor. And so the only thing that can be different is the A is more soluble because it moved farther. And so we would say, okay, A is probably more polar because we had a polar solvent. So A is more polar than B. And sort of a way to test that would be to put them in a nonpolar solvent. So if we took a solution that was nonpolar and put A and B in, and that's true, then B would probably move further than A. Okay? So B would move further than A because now we're in a nonpolar solvent. So B dissolves better. It's more soluble. So B will move further than A because now we're in a nonpolar and B is more nonpolar. Size matters as well. If we had 2 polar molecules, one was really big and one was really small, and put them in a polar solvent, then the small one would move farther even though they both have similar solubilities. So the 2 things we worry about, size and polarity based on, I guess, solubility based on polarity, right? Our solvent that we use as an example is methanol and water. It's considered to be polar, and we're not going to worry about why it's polar. There's something called the dielectric constants and things like that, and you can figure it out. But our solvent's sort of polar. It's not really polar, to be honest, because methanol sort of gets towards that non-polar area. But generally speaking, then, since our solvent is polar, we'd expect polar amino acids to move further. So we looked at, and all the bullet points I just explained, molecular weight and solubility based on polarity as the 2 big factors that we're looking at. So polar amino acids move further in polar solvents, will have a bigger RF value. We'll talk about RF in a second. And polar amino acids in nonpolar solvents will move slower and have a small RF value. Nonpolar would be the exact opposite. Ninhydrin is a chemical that allows us to visualize the amino acids in lab. It's a suspected carcinogen, so we would treat it as being hazardous, and we'd actually spray it in the fume hood with somebody wearing gloves. And then we would leave them in the fume hood when we were done measuring, and then we would dispose of them properly. Sometimes on a plate, you can get an idea of the amino acid just by the color and shape of the spot. And I think one of the that little worksheet I had posted on Canvas, you can kind of see those differences just by the color and shape of the spots in some cases. Not all, but some are very different. The gold standard for determining unknowns, though, is the RF value. And scientifically, it's much stronger to say, oh, these don't look alike, they measure alike, right? So it looks a good start, but being able to say they have the same measurements or very similar measurements is important. So to calculate the RF value, which I've seen 3 or 4 different definitions of what that means. Some people say refractive, relative, relative movement to the front, relative to the spot to the front. So there's a bunch of different ways, but RF, pretty simple. It's calculated by a very simple equation, ds, which is the distance the spot or the sample traveled. So the S you can think of standing for spot or sample, d would be distance, divided by the df. The df is the distance of the solvent front. Okay, since they can't use S because they already used it, right? Solvent, they use F for front. So when the solvent moves up the paper or the gel, we have actually in lab we would use a glass plate that is coated with a silica gel to separate the amino acids. We would allow the solvent to move up, and when we finish the experiment, we would draw a line to show how far the solvent traveled. So it's always relative to the solvent is how far the amino acid itself traveled. Because we're going to divide some number of centimeters divided by some other number of centimeters, a couple things to remember. The DF, right, is going to be the biggest number always. And even if the spot traveled exactly with the DF, it wouldn't be any further in front of it. So you can never have a DS that's bigger than a DF. Okay? So that's number one. Number two, if the spot was totally insoluble in the solvent, right, it wouldn't move at all. So it wouldn't move, it would be 0, right? So you could have a number, let's say the solvent moved 10 centimeters from line to line, where you could have a DS that was 10 centimeters. To be honest, I've never seen it, okay, but theoretically it's possible. So you could have an RF that measured 1.0 exactly. You could have an RF, I've seen this, that doesn't record because there was no movement, so that would be 0 divided by whatever number it was, and that would be 0, okay? Notice that I didn't give any units for RF because the centimeters cancel each other out. But in all other cases, right, you're going to expect some sort of number between 0 and 1. So 0.23, 0.56, 0.89. If you ever calculated 1.27, you're wrong. It's impossible, right? And what you probably did is you probably inverted the DS and DF in your calculation. So you can never have a value that's greater than 1 or negative. You could look in the experimental results and it looks like that, but it's because it wasn't spotted perfectly. You'll never have one that moves backwards. So that's the TLC. For the DNA one, I made it pretty simple for everyone responsible for it. There's two parts to the DNA lab, the electrophoresis and the extraction. And in both cases, there are 4 or 5 questions in the supplement that you just need to answer. And that's what you'll be responsible for. So if you can answer those questions, you could answer any of the questions I would ask on the exam. So you don't need to read that big long thing from Edvitech with the 3' and 5' and DNA and all this other stuff. You just don't have to worry about that. And you know, one of the things that you might want to, you know, think about is I'm not big on procedures generally. Because I think the concepts are much more important than the procedures. You know, one of the questions for the electrophoresis as an example says, well, list the steps in, you know, the electrophoresis process. We're not going to worry about the steps. It's not that important. But the why we're doing it and what's happening, those are really the big important things. Yeah, you need to understand a little about the experiment and how it's set up and how it worked to help understand how the results are relevant. But for the most part, you know, I'm going to concentrate more on the, you know, why do we care? What's the physiological principle involved? Why are we studying this type thing? So don't worry as much about the actual step-by-step procedures as, you know, what's the result of the experiment and how does that have implications physiologically. Those are the big ones. And specifically for the DNA electrophoresis one, I did not ask you that question about list the steps, so don't worry about that one. You can get rid of that one. So for the DNA extraction, I think there are 4 steps. You know, what's in the or 4 questions. You know, why do we use the strawberry? What's in the extraction buffer? Why do we use the ethanol? You know, why do we do this and that? And for the electrophoresis, it was about RFLP analysis and, you know, what things can you use for that test? And, you know, why do we have extra DNA in our genome, basically, and who can have the exact same DNA fingerprints? So those are the questions that you kind of want to look at and pay attention to. Like I said, the number 6 lab is the least represented on the lab quiz, but I'm still pretty sure it's going to have at least 3 points. So that kind of wraps up, you know, a little background of some of the important parts of Lab Quiz 1. Again, you're responsible for everything, so don't just study the stuff I talked about, but certainly know the stuff I talked about, as those are the important things.